Today we're talking about Chapter 11, Section 1, and this is going to focus on evolution at the population level. A population is a group of individuals that are all the same species and live in the same area. So like you would see this group of penguins here on the slide, that would be a population of penguins. Now, genetic variation in our population is important for evolution because remember that is one of Darwin's 4 principles of natural selection. We have overproduction, which means more offspring than can survive. We have variation between those offspring. There are certain traits that are beneficial or adaptations. And then finally, we have descent with modification for that entire species. So we're focusing mostly today on that variation piece. The genetic variation in a population is described by that population's gene pool. And what I mean by gene pool is if we look at all of the individuals within that population and we just make a list of all of the alleles that are in them, those alleles would be our gene pool. And in evolution, that variation in the genes is what leads to variation in phenotype. Remember, phenotype is how those genes get expressed, the physical expression of whatever alleles an organism has. So natural selection is acting on a phenotype, but remember that phenotype is determined by the genes or the alleles present, and so the genetic basis of evolution is definitely very important. When we are talking about genetic variation in a gene pool, one of the major ways that we measure that or quantify that is by counting up alleles and calculating allele frequencies. Allele frequencies, a frequency is just how often a certain allele appears. That can give us an idea of how diverse genetically our population is. So it's just a measure of how common a certain allele is. And we can calculate it if we know the genotypes of all of the individuals in our population. So in this population of frogs here, we have capital G, which is the green allele, codes for green color. We have 7 capital Gs in our gene pool. If we look at all of our individuals, it tells us what their genotypes are. We can count up that there are 7 capital Gs. Because we have 6 different frogs and each frog has 2 alleles, we have 12 total alleles in our gene pool. So the frequency of capital G allele is 7 divided by 12, which is about 0.583. Normally we report frequencies as decimals. You don't really have to convert it all the way to a percentage because whenever you do calculations with a frequency, you are going to want to keep that as a decimal. So we could do the same thing for the brown allele. We count them up, there's 5, so 5 divided by 12 gives us 0.417 as our frequency for the recessive lowercase g allele that codes for brown color. Now let's remember where that genetic variation comes from. This is a review of what we learned in the past. The ultimate source of genetic variation is a mutation or some kind of change in the DNA sequence of a gene. Mutations happen all the time. They can form new alleles, and those new alleles, if they're beneficial, they can be preserved in the population and they would continue. Not all mutations are bad. You can certainly have bad mutations that would cause the death of the individual, and then of course that allele would not continue. But if it is a new variation, like a new color variation that provides a benefit, that allele can certainly continue in the population. We can also have recombination when we remember what happens during crossing over in meiosis. That crossing over process between the chromosomes that you see happening right here, that produces 2 chromosomes that now have a different combination of alleles than those 2 original chromosomes, which means if a gamete is made out of this allele, it is going to look different from a gamete that forms an individual that contains this chromosome that you see over here. So these are the 2 ways that we are going to increase that genetic variation within the members of our population. So what does that mean in terms of actual natural selection? Well, this is in Chapter 11, Section 2. And we know, we've talked about already, that it is populations and not the individuals that evolve. So in looking at the variation within a population, we're going to notice there are certain patterns that can take place. Under a certain set of environmental conditions, one variation might be beneficial, but under a different set of environmental conditions, a different allele or a different variation could become beneficial. So we're going to look at how that can happen, how different environmental conditions can actually cause natural selection to occur in separate patterns. In all of these, we will be looking at a graph, and the blue line in the graph is going to show us basically the distribution of our phenotypes of the original population. And then the red graph is going to show us what that population would look like after natural selection. So in this example here, we have the classic peppered moth example. They show variation between a light color and a dark color and all colors in between with those spots. And basically, the original population of peppered moths was very light with a few dark spots. And then we had the Industrial Revolution. And the Industrial Revolution obviously industrialized 19th century England. They used a lot of coal to power this industry, and therefore there was soot that was on the branches and the trunks of the trees where the moths would live. Well, that changed the environment. The environment was now darker, and it was less beneficial to have this light color, more beneficial to have a dark color. So over time, if you count the number of darker moths, we see a shift in that curve to the right. And so we're going to look at three different patterns. One of them is directional like this, but we'll look at two different patterns that can also result from other ways that the environment can change. Remember that natural selection is acting on the variation. So our bell-shaped curve here, we will start out talking about that because all of these patterns that we're about to talk about are pretty much applying to populations that have a normal distribution of phenotypes. Normal distribution means that bell-shaped curve that you see here. So that means there's very few individuals at either extreme, but here in the middle, whatever that median, mean, average value, whatever that is going to be the one that is most common in a population. You can think about this in terms of human height. If we took all of the students in the school and lined them up by height, most students would fall somewhere in the middle, with very few short people and very few extremely tall people. So this is our assumption for whatever phenotype that we're looking at in the population as it's undergoing natural selection. So this would be our baseline, our normal starting point before natural selection takes place. Our first example of a type of natural selection is directional selection. So in this example, we have one phenotype at one extreme being favored, and whatever's at the opposite extreme is being selected against. You'll hear that term quite a bit. Selected against just means that it's not beneficial, it's not providing a survival advantage, and in fact, it's actually a disadvantage. So we are looking at a Petri dish of bacteria, and bacteria are normally killed by antibiotics. This is a common drug that we would use whenever you have any kind of bacterial infection or a bacterial illness. On this Petri dish, you see those white circles. Those are white circles that are each soaked in a different antibiotic. So the ones that have these clear zones around them are the ones that are effectively killing the bacteria because there's no bacteria growing around that antibiotic. But notice these discs over here, they don't have any dead bacteria. That means the bacteria would be resistant to those antibiotics, whatever is on those discs there. So we have a range of drug resistance in bacteria. It can either have low resistance, meaning it's not going to be able to resist very many antibiotics at all, or it could have a high drug resistance and it would be able to resist a lot of different antibiotics. This right here would be pretty typical. Whatever species of bacteria is growing in this Petri dish, it's somewhere in the middle. It has an average drug resistance. It is resistant to these three, definitely. It's not resistant to those two at all, only a little bit here and a little bit here. So it's somewhere in the middle in terms of its resistance. Well, over time, as we use more and more antibiotics, the environment where these bacteria are living, because of all the antibiotics in the environment, it is going to select for the most resistant bacteria, and the ones that have a low resistance are going to be dying out. So over time, that will shift our curve to the right, and we'll see the mean level of drug resistance is increasing over time. This is an example of directional selection because our curve has shifted in one direction. We have one extreme, high drug resistance, being beneficial. The other extreme of low drug resistance is being selected against. Our next example is called stabilizing selection. And in stabilizing selection, now we have selection for that mean phenotype or an intermediate phenotype. And now both of the extremes are going to be selected against for whatever reason. So our example that we have here, there is a fly. This is the fly. It lays its eggs in this goldenrod plant here and produces a swelling on the stem called a gall. So the baby flies are inside of that gall. Well, if the gall is small, then the wasp, this is a parasitic wasp, what it does is it finds these galls on the goldenrod plant, it lays its eggs inside of them, and then the wasp larvae will eat the fly larvae. And that obviously is bad for the fly, because if its larvae are being consumed, then its population size will be decreasing. On the other extreme, if it has a very large gall size, birds like the woodpecker are very attracted to that to eat it as a food source. So if the gall is really large, then it is going to be eaten by a predator. So we have a situation where a small gall size is bad because of the parasites, but a large gall size is bad because it will be eaten by predators. So that is selection that is going to favor this intermediate gall size, or a size that is somewhere in between those two extremes. That is going to make our curve look taller and spikier and less wide and rounded compared to that original bell-shaped curve that we had in the population. So again, this is an example of stabilizing selection, and it occurs when the intermediate phenotype is beneficial, but both extremes are being selected against. And finally, our third pattern of natural selection is called disruptive selection. Disruptive selection is exactly the opposite of stabilizing. In this graph, you see the dashed line shows the mean population, with our intermediate phenotype being the most common, as we would expect. Well, if the mean phenotype, whatever this is now being selected against, its numbers are going to decrease, and both of the extremes are going to start to increase. So you get this shape in the curve that looks more like an M than that bell-shaped curve that we saw at the beginning. Your example here is in the birds that we see on the screen. These are buntings. Males can have two different colors. They are either more brown in color with just a little bit of blue, or they are this very bright blue color. So in the bunting population, these brown male buntings, they have a survival advantage because they don't stand out. They're more camouflaged, but they get fewer mates because the female birds prefer the blue color. So there is an advantage. There's a survival advantage to being brown. So that extreme does actually get selected for. The other extreme, the blue extreme, gets selected for because the female birds choose it more often when mating. So that's our beneficial on the blue side. And then if they're somewhere in the middle, if they're half brown, half blue, they're too blue to get that benefit of the camouflage, but they're too brown to be attractive to a female. So the mean or that combination of brown and blue is being selected against because there's less chance of survival and less chance of mating, so those numbers would go down. So hopefully you found this to be helpful, and if you have any questions as you look over your notes, please make sure that you ask them in class.